Enzyme-linked immunosorbent assay, ELISA, is one of the most commonly used techniques in biomedical research and medical diagnostics. It harnesses the specific interaction between antibodies and their respective antigens. It can be used to detect a specific antibody or antigen in a biological sample. Suppose you want to determine whether a patient has recently been exposed to a certain virus. You could take a serum sample and test it for the presence of antibodies against the protein coat of the virus. How are you going to distinguish this antibody from all the many other antibodies and serum proteins in the sample? You'll have to make use of the one feature that makes this antibody unique, its specific binding to its antigen, the coat protein of the virus in question. In other words, you're going to set a trap using the antigen as bait. So you take a simple plastic well and coat the bottom of the well with the antigen in question. You'll also need to add a blocking solution made of proteins that do not form any specific interactions with the components of the serum sample that you're testing. Now you add in your serum sample. If the target antibody is present, it will bind firmly to the antigen at the bottom of the well. The blocking proteins ensure that no other proteins in the serum sample will bind non-specifically to the inside of the plastic well. Then you wash out all the proteins that didn't bind. Now, if the antibody were a macroscopic object visible to the naked eye, it would be pretty easy to tell if it was present in the sample. Obviously, antibodies are too small to see. So how are you going to work out whether it's there in the well or not? The solution is to come in with a second antibody that is conjugated, that is attached to an enzyme called a reporter enzyme. A typical reporter enzyme is HRP, horseradish peroxidase. HRP catalyzes a color change reaction. In the example shown here, the antibody you're testing for is a human IgG, so the secondary antibody needs to be able to recognize and bind specifically to human IgGs. These antibodies are commercially available. They bind to the invariant region of the IgG, whereas the antigen binds to the variant region. After washing away any unbound antibody, we add the substrate of the HRP enzyme to the well. If there is no HRP in the well, the solution remains colorless. If HRP is present, the substrate is converted to a colored compound that is visible to the naked eye. Thus, you can easily distinguish whether the original serum sample contained the antibody of interest. The type of ELISA described here is called indirect ELISA because the reporter enzyme is not directly attached to the antibody that recognizes the antigen in question. As we have just seen, indirect ELISA is used to determine whether a specific antibody is present in a sample. The ELISA technique can also be used to determine whether a specific antigen is present in a sample. The simplest way to do this is direct ELISA. In direct ELISA, the serum sample itself is coated onto the plastic well, so that the various antigens in the sample adsorb onto the well and are immobilized. To detect your specific antigen of interest, which could be a viral protein or a cancer biomarker, for example, you simply add in an antibody that binds specifically to that antigen of interest. This primary antibody is conjugated to the reporter enzyme HRP. As before, when unbound antibody has been washed away, the substrate of the HRP enzyme is added, and a color change reaction indicates a positive result. So in direct ELISA, the reporter enzyme is directly attached to the antibody that recognizes the antigen. The problem with direct ELISA is that there is a high risk of non-specific interactions because all the proteins in the sample remain in the well. And the sensitivity is quite low because there is no amplification of the signal. One technique with greater specificity and sensitivity is sandwich ELISA. The aim of sandwich ELISA, like direct ELISA, is to detect an antigen of interest. In sandwich ELISA, the well is first coated with an antibody that recognizes the antigen of interest. This is called the capture antibody. Next, the sample is added to the well. The antigen of interest is captured by the antibody, and the rest of the sample is washed away. Now, a second antibody is added, one that recognizes another epitope of the same antigen. The antigen is thus sandwiched between two different antibodies. The second antibody is either directly attached to a reporter enzyme, as in direct ELISA, or indirectly attached to a reporter enzyme, as in indirect ELISA. Either way, when the substrate of the HRP is added, a color change reaction indicates that the antigen of interest was present in the original sample. So the three main types of ELISA are indirect, direct, and sandwich ELISA, and they all make use of the specific interaction between an antibody and its antigen in order to detect a protein of interest in a complex mixture. In practice, ELISAs are usually performed in 96-well microtiter plates. 96 wells might seem like a lot, but you often need to test many samples at once, or to test different dilutions of each sample, and furthermore, you always need to have positive and negative control wells to ensure that the test is working. ELISAs can be performed manually by making additions with a multi-channel pipette and removing washes by flicking the plate gently over the sink, or they can be performed by an automated ELISA workstation. ELISA results can be qualitative or quantitative. If you simply want to know if the target protein was present in a sample or not, you can look at the plate by eye. However, if you want to determine how much target protein was present, an ELISA plate reader can perform a spectroscopic analysis of each well. By comparing the coloration of a well to a known standard curve, it is possible to calculate the concentration of the target protein in the original sample. As well as detecting antibodies and disease markers in serum samples, ELISAs can detect pregnancy hormones in urine, or cytokines in cell extracts, or allergens in food samples, among many other things. Enzyme-linked immunosorbent assay is a versatile technique whereby the specific antibody-antigen interaction is harnessed to detect the presence and/or amount of a target protein in a biological sample.